Hi, everyone. I hope you hear me well. Thank you so much for connecting today. It's a pleasure for me to take part in this Global Young Scientists Summit and to introduce you this session today, hosted by Prof Virgile Viasnoff. We'll talk to you about organoids and more particularly why they revolutionize biology and medicine, all the while posing some challenges even outside the life sciences field. But first of all, let me introduce you quickly the CNRS, which is the French Center for Scientific Research, which has been established in ASEAN since 2007 through a first representative office in Vietnam before moving to Singapore in 2014. CNRS has now 4 international research laboratories in Singapore: IPA, which is working on AI, Sintra, which is working on nanomaterials, Majulab, working on quantum technologies, and finally BMC2, working on mechanobiology. And in addition to these, 2 other international labs are located in Thailand and Vietnam, and several, albeit a bit smaller, projects are ongoing in most of the ASEAN countries as well. And in addition to this representative office, CNRS recently joined the CREATE ecosystem in 2019, and this is the first subsidiary that we have abroad, CNRS at CREATE. And CREATE itself is an international collaborative initiative in Singapore, housing research centers set up by the top global universities, such as the MIT, Cambridge, or even the Technical University of Munich. And currently, CNRS at CREATE hosts or is part of one CREATE program, which is a Descartes program, 2 seed collaborations, Bubble and Into-Make, and 4 intra-CREATE projects, namely EcoCities, ScanCells, Space, and Calypso, which stands for Computer-Assisted Live Imaging for Predictive Screening of Organoids. And this project started in October 2020, and it's in collaboration with CNRS at CREATE, NUS, and NTU. And its lead PI is Prof Virgile Viasnoff, who is here with us today. And he will tell you a bit more about these initiatives in particular in a moment, I'm sure. And in addition to this project, Virgile is also the director of the CNRS IRL BMC2 that I mentioned a bit earlier, and also the co-founder of 2 startups, Pixel and Reflect. And to tell you a bit more now about his own research trajectory, Virgile was recruited by the CNRS at ESP-CI in Paris in 2006, where he worked on single molecule biophysics. And he then joined MBI, the Mechanobiology Institute of NUS, in 2010 as an associate professor working on cell-cell junction mechanics and microfabrication of pattern substrate for cell culture. So now I think, without further ado, I leave the floor to you, Virgile, to present a bit in more details what you do.
Okay, thank you very much, Celia. Thank you all for participating here. Let me share my, no, you see my video already, I guess, yes. So what I'd like to do today is share with you my enthusiasm about organoids and try to convince you that these systems, these new systems in biology, not only need strong biologists, but also all kinds of scientists, including computer scientists, including physicists, including bioengineers. And I'll, for a moment, in a moment, I'll show you why. But I guess many of you, if not the vast majority of it, may not have heard about organoids, but I'm sure you're way more familiar with this sort of sci-fi scenes where you see a little blob of, or someone being cut an arm and the arm grows back, or someone, a little blob of cells growing back into the full body of usually, I don't know why, the supervillain. So that sounds quite sci-fi-like. But unlike other sci-fi dreams, the idea that you can regenerate limbs or you can regenerate a full body just out of the blue like that is not science fiction at all. So that happens in life. You have here the regeneration of a limb in newts or tritons. You see this is an amputated limbs here. This film is from the HHMI Institute. And you see that when you cut it and you let it evolve, the newt has this ability to grow back the exact original limb as when during the development of the embryo. So anything that is not completely, that is not prohibited by the laws of physics might certainly be possible, even though people have dreamt it in films. So this is, still with not really enriched, but if you imagine that you can actually get some cells out of your body and regrow an organ, either a full organ or part of it, and you can imagine the potential of that for biology. You can try to understand much better how the development, how regeneration happens. You can use it as a regenerative medicine and reimplant it into your body. You can use it as a way to test drugs and so on and so forth. So let me tell you a little bit where the field is, and I'll be obviously sketchy in these 20 minutes, but I hope I will give you some interesting landmarks for you to be convinced that you can come and work on organoids field. So, one of the historical hallmark is when there was this realization that if you take cells from a patient or from anyone, and depending on how you grow them, if you grow them in 2D or in 3D, the cells will behave very differently. And that is the main work of Mina Bissell with some sort of landmark publication in 1987, where they show that ex vivo, when you take the cells out of your body, you can grow them not in 2D, but in 3D, in different matrices, in different medium. And when you do that, the cells will behave differently. And in her case, it was breast cancer. They will be either cancerous or kind of benign, depending on how you grow them. So that points out to the very important role of microenvironment and the importance of growing cells in 3D rather than in 2D. Now, another hallmark in the field was, of course, people knew about stem cells and the fact that you have cells that are stem. In other words, they can differentiate and they can become any cell in your body afterwards. But the question was, how do you get these stem cells? And usually at that time, the stem cells were coming from embryonic bodies or, umbilical cord at best, and that posed ethical problems. Now, in 2006, there was this discovery of what is called induced pluripotent stem cells, where actually the Shinya Yamanaka, showed that you can actually take somatic cells, which means cells which are already differentiated cells from your skin, pretty much. And using a combination of growth factors and chemicals, you can reprogram them to be stem. In other words, you can take cells from your skin, reprogram them to be stem, and then push them back to become either neural, skin, pancreatic, blood, adipocyte, different kinds of cell lineage, which means now that, if I want to be sketchy, I can take really cell, again, from skin, for example, because they are easy to access, and then take them into and try to redifferentiate them into something that looks like cells that you have in other organs. So that's opened the possibility also to try and grow organs from your own cells. Now, the last historical hallmark I want to tell you about is in 2009, in the lab of Hans Clevers, they show that you can start here, with a tiny aggregate of intestinal stem cells, so stem cells that reside in your intestine, because they find a marker for them, and then let them grow in 3D in this gel, like Mina Bissell have done, and then let them grow over days and days and days. And rather than just being completely growing as a sphere in a really disorganized fashion, over time, they would develop crypts and cavities which resemble, not only morphologically, but also in terms of the cell composition inside, they resemble your intestine. Okay, so that was the first example where you can take stem cells and grow then a mini organ in a tube, if you wish. So that is sort of historically the launch of what is called organoids, and organoids are thus these ability for you to take cells from your body, reprogram them, and then by a series of chemical treatments, you can grow them in tubes and make them create structures that either mimic the full, well, not really the full, but usually a part of your organ at the moment. Okay, and with that, you can create any kind of organs. And pretty much every other month now, or every 3 months, there is a new organ that where people claim that they have differentiated their cells in, and they create an organoid of. So let me give you some example. I'll start with an example that I like because it's been developed in Singapore. There's no order of it's only order of my preferences based on my knowledge of them to some extent. So this is a kidney, a mini kidney, if you want, grown from stem cells in the lab of sorry. Yes. Okay. All right. Of Yunja at NTU. And you see these beautiful branches coming out of this ball of cells, which mimic what happens in the nephron of your kidney. This thing is huge. It's about 1 millimeter in diameter. So they started with a few cells and they let it grow all the way to millimeters to have these beautiful structures. Now, one of the most fascinating organoid also to date is the brain. So the brain cells, people take, again, stem cells and differentiate them in neurons and let them grow. And the neurons differentiate in different kinds of neurons and organize in space. And you have here an example of just an image of an example of brains. Now people can grow these mini brains for years, literally. Right, and in the lab of Madeleine Lancaster, they even connected these mini brains to the spine of a mouse and see that the spine has spontaneous motion linked to the firing of the motor neurons into the muscle. So you grow these guys, literally for almost centimeters for the brain. Give me another example, which is now you can also take the very primary cells, embryonic stem cells, and then let them develop into structures that somehow mimic the embryo development. So they are called gastroloids, and you see that these stem cells, they can grow in large bodies like that, which mimics the embryo development here, and then later on they develop different kinds of shapes. But not only do they have the right shapes in terms of organization, but depending on the treatment that you put, you can start having cells that form a heart in them, or muscles, or even muscles and gut. So pretty much you're mimicking in the test tube how the specific lineages, you're targeting specific lineages of cells that lead to the development of organ-like structures here in embryo. Okay, and I can go on forever like this. You have now here you can have the retina that has been developed. You have pancreas. You have here just a mini heart. You have here a liver, and so on and so forth. So don't get me wrong. At the moment, and that's where I want to come, is it's not developing really full organ. It is miniature organs which recapitulate some of the cell arrangements, but they may not be always functional and hardly functional in test tube. But you can take some of them, especially liver, and regraft them into the mouse liver and help. And that mini organ would grow inside the mouse liver and sort of regenerate the liver and cure it from different diseases. So what are the challenges here? Of course, there is an enormous amount of challenge from the biological part, which is how do you get these cells to differentiate? How do you get them to properly form the structure? But there is another thing that happens is when the organ they develop in a very stereotypical manner. They are very well organized, and every individual has the organ organized pretty much the same way. However, when you grow these organoids in tubes, they are not provided with any scaffold. They are not provided with any time-dependent guidance from the development of surrounding organs, and hence there is a very large variability in the way they develop in terms of morphology, in terms of the type of cells that are differentiating. So what happens when I show you those beautiful images is usually you have many organoids developing, and then you pick the ones that develop like the organoids in the like the organs, sorry, in the body. So the question there is the following: if you want to use these systems in order to do, let's say, transplantation, well, you must be sure that you're picking the rightly developed organoid to retransplant it. You don't want to transplant something that is faulty. If you want to use drug development, you have to control the variability of these organoids in order to be able to compare how the effects of drugs alter the different organs in vitro. Of course, you want also to develop biological model out of that. So you use these organoids in order to understand how the biology happens, how the organ develop, because now you have direct access to these to the steps of development, and you can even stop them. You can even look at them one by one. And so on and so forth. So you have many things that you can do with these organoids. But to do that, you have four things that you have to masterize. Engineering of the cell, and that's to some extent the work of biologists, where you want to design the cells, you want to provide the cells with the proper growth factors in order to be able to reprogram them stably and to develop the right organoid. You have other problems that are linked more to engineering or bioengineering or chemical engineering. So you have to engineer also the environment that surrounds this organoid, way more than we can do now. And that is prominent if you want now to give a clear path for development. Now you can also enhance the context. So in other words, how do I provide the flow and how do I exchange the medium above the cells and above the system? So here you have problems for engineers. And then you have here enhance of readout. How do I image? How do I know my cells are properly developed? How do I treat all the data coming from this organoid? And you have immense problems here, especially adapted to AI. And that's what I want to come to in the next 5 minutes to present you the project Calypso, where you have all the partners of the project here. Pretty much the idea was to develop the high content imaging platform for organoids. And I won't go into the details, but the idea was to try to, at the same time, get the organoid into tiny micro niches to control the environment around them, and at the same time enable their rapid imaging. So the way we did it, we created these micro niches by creating cavities which are inverted truncated pyramids into which we seed the cells. And once the cells are entering the pyramid, the edge of the pyramid, the faces of the pyramid are reflective. And so we can use them as a mirror to now see and illuminate my organoid on every side at the same time, which allows very fast imaging of the organoid. And why do we want to do that? We want to look at the structure of the organoid, either fixed or live. So the way you do it is you have your organoid there, you move a laser without moving anything else along the mirrors, and as you move the laser along the mirror, you can scan, and within 1 hour, you can image up to 100 organoids automatically detected because they are arrayed, and you can do it in 3D, 3 wavelengths, and that takes a reasonable amount of data. So once you have all these data available, you see that because you can collect so many, the idea is to tackle on the large number of data that you have in order to be able to image many organoids and use AI to characterize their morphologies. And that's what we did. We cultured many organoids, many kinds of organoids, and we developed generic AI systems to measure the position of every cell, the stage of development of every cell, the stage of division, of every cell, to know if the organoids are viable, as well as looking at live imaging. And here what you're looking is 2 or 3 hours 2 or 3 days of development of an organoid, where when the cells change color, they change fate. So this is, for those who know, stem cell going into endoderm, and we can measure now how the development occurs when you have these stem cells being differentiated within the ball of cells, and that poses a lot of problems, a lot of questions in terms of physics that we can now address in terms of how do you have segregation of tissue in a very standardized manner. the idea behind the project, and I'll leave you there, is now if you have a large collection of images, you can create large libraries and use this AI to create a multiscale analysis of the morphologies of the organoid in order to quantify their variability. And that creates database of quantitative assessment of the heterogeneity, which then now you can use different drugs or different treatments to assess the changes in variability. Now, the more I'd say daring perspective is actually to do the live imaging, and from the live imaging try to use AI in order to actually create a predictive map of where the organoid, how the organoid will develop. So in other words, if I see how my organoid develop, can I predict in 2 days what state it will be in, or whether it will be properly properly shaped, or developing the right stem, the right cell types, or will it completely be off track? So that allows to address many problems, including these problems of developmental path, as well as the ability to do quality control on the quality of the organoid development. So with this, I'd like to stop here. Thank, of course, all the people who have been working in this project that has been a way or another ongoing for the last 8 years. And just tell you that if you're interested in organoids, this is a wonderful field. You should definitely come and help, whatever your specialty is, because there is a need for pretty much every single expertise in this field. And with this, I thank you very much. All right, so let me maybe take the questions now. Yeah. Okay. So I'll take the first question. I see one on the chat. How do you see the possibility of growing brain cells to better understand brain diseases and help recover the connection between the brain to the spinal connection? Yeah, so, you know, the ability to, well, the question you ask is not only for brain, but I think it's general. So the brain organoid has been also one of the first ones developed, and it is indeed what people use it to see the development of brain, and especially human brains, because here you can definitely image the organoids from human stem cells, which is usually hard to see the development of human organs. That is absolutely what you're saying is trying to understand better neurodegenerative diseases and so on is exactly the direction that the people working on brain organoids are taking. I don't know. I see all the questions have just disappeared for some reason. So can you just type them again on the platform? Yes, Celia.
I think the session has ended.
Okay.
So thank you very much.
Okay.
I think the time was a bit short.
Okay.